All right, so what we're going to do is we're going to finish up the notes on the sugars that were made in the Calvin cycle. So what actually happens to those triose phosphates? So remember, those were 3-carbon sugars. So really two things happen. They either become starch or they become sucrose. And we're going to talk about why they might become starch or sucrose in just a moment. So first we're going to start with the starch synthesis, and that's what we see on this particular page. So with starch synthesis, what we really need is we need two things. We need something called ADP glucose, that's glucose with ADP attached to it, and then we need starch synthase, and that's going to be the enzyme that's going to catalyze this reaction. I do want to notice that there are two reactive groups, so there's XA and XB, and so what's actually occurring here is that they're going to alternate between the two of them, adding on the glucose molecules to form the starch. And so let's just kind of squeak ahead here. XA, right, is going to add the third glucose, and XB is going to add the fourth glucose, and then XA is going to add the fifth, and XB is going to add the sixth, so on and so forth. And so they just alternate between the two of them. So what is ADP glucose? It's just an activated nucleotide sugar. And really how we get this is it's a condensation of glucose 1-phosphate with ATP. And whenever it's made into ADP glucose, it's an irreversible reaction. And so what's going to happen then is that starch synthase then transfers that glucose residue from ADP glucose to the pre-existing starch molecules. So you can see here, as we look in the end here, that 2 build onto 1, 3 build onto 2, 4 build onto 3, so on and so forth. Glucose is added to the non-reducing end, so we have the non-reducing end here, and then we keep adding. Just like in glycogen synthesis, when we talked about that earlier in the semester. So again, just two active sites, we attach one and then the other, and this increases the effectiveness or the efficiency of this. So that's starch synthesis. The next one is sucrose synthesis. So for sucrose synthesis, again, we're going to have an activated glucose. This time it's UDP instead of ADP, so it's a UDP glucose molecule. And the other important component is fructose 6-phosphate. Additionally, I apologize because on your outline, it doesn't have the two enzymes that are required. So we have sucrose 6-phosphate synthase and sucrose 6-phosphate phosphatase. And so hopefully okay, you see synthase, it's going to synthesize or condense, bring two things together. In this case, it's going to be the glucose and the fructose 6-phosphate. And then what's a phosphatase do? Oh, well, it removes an inorganic phosphate, so now we're going to have our final product. So let's look at this particular one again. So UDP glucose, right, so it's a glucose molecule with, again, that activated nucleotide on it. And what this is going to do is it's going to combine with the fructose 6-phosphate. You might ask yourself, where do we get these? Well, remember that this is what's happening to the triose phosphates. So, for instance, the fructose 6-phosphate molecule, that's going to form after we condense two of the triose phosphates together. They're going to form the fructose 1,6-bisphosphate. Boy, this sounds familiar, which was catalyzed by aldolase, and then it's going to have a hydrolysis of it, and we're going to yield the fructose 6-phosphate. So that's just it. For instance, you don't have to know the details of that, right? But those trioses are going to come together to form our starting components. All right, so we have our starting components here, and then what we're going to do is the synthase is going to bring it together. We're going to remove that UDP. We're going to have this linkage between the anomeric carbon here, carbon 1 and carbon 2. What's interesting about this particular one is that then we get the sucrose 6-phosphate. We're going to cleave off that phosphate using a phosphatase, and we get sucrose. So why sucrose? So this was the question I kind of asked at the beginning. Why are we creating sucrose? They believe that sucrose may have been selected as the transport form of this carbon because of this unusual linkage. And the unusual linkage is between that anomeric carbon 1 of glucose and the carbon 2 of our fructose. And this is the anomeric carbon of fructose, so it's between two anomeric carbons. And this bond is not hydrolyzed by amylase. And amylase is a common carbohydrate cleaving enzyme. And so because amylase can't cleave this bond between the anomeric carbons, it's going to prevent it from reacting to non-enzymatic enzymatically. So glucose will react with amino acids and with proteins, but sucrose doesn't. Okay, so sucrose doesn't have these kind of off-target effects, and so it's going to be transported better through the system. All right, so then kind of the last note for this semester, wow, can you believe that, is starch versus sucrose synthesis and its regulation. So when we're looking at that, there's really two components that are going to regulate starch and sucrose synthesis. And the first one is the amount of inorganic phosphate, so RPI, and then the amount of our triose sugars. And so when we have a really high level of cytoplasmic inorganic phosphate, our triose phosphates are going to be exported in exchange for our inorganic phosphates, and it's going to be used to synthesize fructose. So this is going to activate this pathway, and we're going to get sucrose. However, if we have really low levels of our inorganic phosphate, then the triose phosphates are going to be retained in the chloroplast, and it's going to be used to synthesize starch. The other component that I wanted to talk about, so phosphorylation obviously is going to play a very important role here. But the other component is the fructose 2,6-bisphosphate. Oh no, it's come back, right? Fructose 2,6-bisphosphate, because that was an important regulator of another pathway that we've already gone through. And so this particular fructose 2,6-bisphosphate, as you can see itself in the dark reactions, is going to activate a kinase 1 that's going to allow us to get our fructose 1,6-bisphosphate, which is going to allow us to get our triose phosphates. Right, and then also it's going to regulate this component, so it's going to stop our sucrose as we go through. The other component that I wanted to talk about is our phosphatase, so phosphorylation. So there is a kinase and there is a phosphatase that is that helps regulate our sucrose level. And so the protein kinase is specific for, it's called SPS, which just stands for sucrose phosphate synthase, right? And so it's going to phosphorylate a serine residue and activating it, and a phosphatase is going to reverse this inhibition, and it's going to help regulate this concentration as we go through. All right, so that's pretty much the end. I do want to say that there is a balance between, our triose phosphates are really produced by the Calvin cycle when we have bright sunlight, because remember, we need that energy from the light, but they may be stored temporarily in the chloroplast as starch, or they could be converted to sucrose and exported to non-photosynthetic parts of the plant, or it could be both. And so the balance between these is regulated and they're coordinated so that we have the correct rate of carbon fixation. And that is the last note for our class. So I will see you guys on Wednesday.